And welcome to the cellular respiration and fermentation lecture. I'm just going to be talking kind of about an intro to the different reactions that are occurring and the different steps. So remember that life needs to do work, and work is energy. Energy is the ability to actually do work. And life all started in the stars and fusion reactions occurring, and all of life on Earth depends on light energy coming from the sun, and with that we're able to take that light energy and convert it into glucose and photosynthesis, and then we're able to create ATP and cellular respiration. So we need to talk about what an oxidation and reduction reactions are. So remember that compounds that participate in an exergonic reaction can act as fuel. So pretty much we're going to be breaking down glucose, which is going to be an exergonic reaction to create ATP. And then we are going to be taking ATP and also breaking off those phosphate groups to release energy. So both are exergonic reactions. And we're going to be using many different enzymes to break down this complex molecule into our potential energy. Some is used to do work, we store some, and then some is lost as heat. So there's different types of ways that we can create ATP energy from food. And one is called fermentation. Fermentation does not require oxygen to occur. We see this a lot of times in different organisms that may not live in an oxygen-rich environment. We also see fermentation occur when animals do like a fast or very strenuous motion. So for example, if you are doing like a long distance sprint and you're sprinting as fast as you can, a lot of times you're going to go through fermentation, which actually builds up lactic acid in the muscles, and that's where sometimes you get muscle soreness from. Then you have aerobic respiration. Aerobic respiration is going to need oxygen, and if you remember, oxygen and glucose are what are required in order for us to go through cellular respiration completely. So oxygen is going to be required for aerobic respiration. And cellular respiration is going to include both anaerobic and aerobic respiration, but it's very optimal when we have the oxygen involved. So the overall process can be demonstrated in a simple equation. Our organic compound, which is going to be glucose for the most part, and oxygen. And then our products are going to be carbon dioxide and water, which are considered our waste products, and then our energy. So chemically, it looks like this. Glucose, C6H12O6, plus 6 oxygen is going to come out to be 6 carbon dioxide, 6 water, and then energy. Remember that this is going to be a negative delta G, and it's going to take approximately 686 kcals to complete that chemical reaction. So now we're going to get into what's called a redox reaction. So a redox reaction also is known as an oxidation reduction reaction, and this is basically when we're transferring one or more electrons from a reactant to another. So whatever loses the electrons, or the loss of electrons, is called an oxidation. You're oxidizing it. And the addition of the electron to the other substance is called reduction. So that's why it's an oxidation reduction reaction, because we're actually taking these electrons and then transferring them to different products. And you can see here that this actually lost the electron, and this one gained the electron. That's why it's got the negative charge. So the substance that donates the electron is going to be known as the reducing agent. So the reactant that actually donates the electron, and the reactant and then the product that accepts the electron is going to be known as the oxidizing agents. And remember that not all the time are electrons just stripped and moved, but they can be shared between a polar covalent bond. It does take much more energy to actually remove the electrons. So we're going to take a look at an example here of a redox reaction. And what I want you to notice here is our reactants. So what we start with is methane and oxygen. And what we're going to convert it to is carbon dioxide, energy, and water. So what we say is we say that the methane it went through oxidation. And what do we mean by that? So methane is going to be the reducing agent because it's actually losing some of its electrons to the more electronegative carbon dioxide. And now these are being shared unequally. And then looking at the O2, it shares the electrons equally, but once it reacts with the H in the methane, it actually gains electrons and is now going to be the electrons are now going to be pulled closer to the oxygen atoms. Okay, that's pretty common. We know oxygen is more electronegative than hydrogen. So O2 acts as our oxidizing agent. So when looking at the cellular respiration equation, which is the glucose, the oxygen turning into your carbon dioxide, water, and our energy, glucose is going to be your reducing agent and oxygen is going to be your oxidizing reagent. Glucose is an excellent fuel because it has tons of hydrogens with bonds as a source for basically hilltop electrons. And what this means is these electrons that are attached to the hydrogens are going to be really easily transferred to oxygen to make a water molecule. So it's really easy to kind of manipulate these electrons. And as we get in all the processes of cellular respiration, what you're going to see is the harvesting of the electrons from glucose is important. The electrons are where we actually really get the energy to make the ATP molecule. And glucose has a lot of these electrons that are very, very easy to harvest. So cellular respiration doesn't just do one step and boom, we have ATP. It wouldn't be as efficient if we did that. A great example of this is, let's say you're driving and your gas tank explodes, you're not going to get very far. You're probably going to be able to drive really fast for a really short amount of time, but it's not going to be very far. So that's why when we harvest these electrons and oxidize glucose, we actually go through many steps so that we're able to really get as much out of it as possible. And each step in cellular respiration is going to use a specific enzyme that catalyzes the reaction. And it's really cool to look at it all in big picture. It is out of the scope and sequence to memorize all of these different enzymes. So don't necessarily worry too much about knowing all of that. And in some steps, you're going to directly see an electron stripped from the glucose in the form of a hydrogen. You're going to have an H plus atom, and then you're going to add a hydrogen to it. And it's through these really two cool molecules called NADH and FADH2 that we're going to get into. And these molecules here are what are going to accept the H, the hydrogen that's carrying those electrons. So how does NADH or NAD plus actually get the electrons from glucose? So enzymes are going to remove a pair of hydrogen atoms. And remember, with these hydrogen atoms, you're going to have 2 protons and 2 electrons. So the 2 electrons and the proton are going to attach to NAD plus. And this is going to form what we call NADH. The other proton, okay, that we saw up here, the other proton, is going to get released into the surrounding solution as basically just a H plus ion. So we're harvesting we're changing this NAD plus right here, this molecule. We're adding 2 electrons to it, 1 proton, and 1 hydrogen. So that's what allows it to now become a neutral atom. And then this other proton and other hydrogen we had are just going to be floating around in the cytoplasm or inside of the mitochondria as an H plus ion. So if we look here at how this actually harvests the energy, we have this really cool electron carrier, which we call a coenzyme, which is going to be that NAD plus molecule. And it's really cool how this happens because, like I said, we take our two hydrogen ions with our two electrons, and we're able to change that NAD plus to an NADH molecule. And right now you're maybe a little bit like, I don't understand why this NADH is so important. But once you start looking at all the steps of cellular respiration, this molecule right here is what takes the electrons that we've harvested from glucose and actually allows them to become an ATP molecule. So how did the electrons that are stored in NADH finally become a water molecule? Because this hydrogen right here is going to become our H2O molecule that is our waste product. So how do we harvest these electrons, or how do we get these electrons from this NADH molecule to make ATP and make our H2O molecules? And that's called the electron transport chain. And we're going to get into a lot more details about what this electron transport chain is. Just right now that it's built on the cristae of the mitochondria, so where the folds in the inner mitochondrial membrane are. And the electrons are moved there in this NADH molecule. And what happens is we use the electrons to basically kind of activate each enzyme in each step, and we use it to pump hydrogen ions across that inner mitochondrial membrane, and that those H plus ions are what's going to actually go through ATP synthase and then create the ATP molecule. So we're going to go ahead and stop here because I want you to watch a few videos on the NADH molecule so you can get a better idea. I know this is a ton of information at first, but just know you're going to get a lot of practice with this, and you're really going to be able to understand it in depth.